Digestion and absorption are the ultimate functions of the GI tract. The majority of nutrient absorption occurs in the first portion of the intestines. The thickness of these arrows shows the magnitude of absorption at each site in the intestine. So most of the nutrients are absorbed in the duodenum, some in the jejunum, and less in the ileum. The colon is then primarily responsible for the final removal of electrolyte and water, although there's also absorption of some short-chain fatty acids produced by colonic bacteria. The small intestine has some specialized structures that increase the absorptive surface area of the mucosa to facilitate this absorptive process. And this image shows each layer of these specializations. So there are folds in the mucosa itself, and these folds increase the surface area around threefold. Then the mucosa has its own finger-like projections that protrude into the lumen, and they're called villi, and those further increase the surface area another tenfold. And then each individual epithelial cell on this surface of this villus has microvilli, and those microvilli, or brush border, increase the surface area another 20-fold. And this microvillus surface is sometimes called the brush border because it looks like the paintbrush under a microscope. The overall increase in surface area here as a result of all of these levels of specialization is somewhere around 600-fold. In this view of the intestinal mucosa, it's easier to see the epithelial layer and the individual cells of the epithelium, as well as the blood vessels and lymphatic or lacteal that are in the lamina propria underneath. So there's a gradient of cell function of these epithelial cells in the intestinal epithelium. The cells of the villus, as we've talked about many times, are absorptive cells, and they primarily absorb nutrients from the lumen, while the cells of the crypt are primarily secretory cells. So if we look here, and we'll zoom in on this box here of the cells of the lumen, there are nutrients in the lumen, and they're digested from their large ingested form into smaller absorbable molecules with the help of secreted digestive enzymes as well as brush border enzymes present on these enterocytes. And these nutrient components, when they're broken down, can then be absorbed from the lumen across the epithelial cells and into the blood in the case of amino acids and monosaccharides, or into the lymph in the case of lipids for distribution throughout the body. So absorption of nutrients across the mucosa can be either by a paracellular route between the enterocytes, or by a transcellular route across the enterocyte. So if the lumen is out here and the blood space is out here, the paracellular route is passive, and this depends on the leakiness of the section of the intestine. So that's how tight those tight junctions are that link the epithelial cells. So the duodenum is the most leaky, and the small intestine epithelium becomes tighter as you move more distally. The transcellular route can be either active or passive movement of nutrients or ions, and it occurs through several mechanisms, and generally this relies on the presence of specific transporters present on the luminal and then basolateral membranes to get the nutrients out into the blood. So this includes things like ion channels or transport proteins, as well as exchanger proteins. And throughout the GI tract, as we'll see, the absorption of water is primarily through the paracellular route by osmosis. So the water follows solute.